Welcome to the Sports Science Dudes. I'm your host, Dr. Jose Antonio, with my co-host, Dr. Tony Ricci. If you're a first-time listener, hit the subscribe button and like the show. You can find us on YouTube, Rumble, Spotify, and Apple Podcasts. Our special guest today is Dr. Sara Campbell. She is an associate prof in the Department of Kinesiology and Health at Rutgers University in New Jersey. Tony's, I don't know if Tony loves that state. It's sort of.
I like it more than New York, actually.
Yeah, well, I think we all like anything more than New York. She got her BS and MS from Bloomsburg University in Pennsylvania and her PhD at Florida State University. Following her PhD, she did a 3-year postdoc. I did not know this. She did it at the USDA, which is interesting. You investigated the ability of flaxseed, and this will be interesting because I don't know anything about flaxseed. Looked at the ability of flaxseed to reverse atherosclerotic lesions. And at Rutgers, you do a whole bunch of stuff. The Campbell Lab investigates the intersection of sex, diet, and exercise on gut microbiota, which you are going to educate Tony and I on.
Very much.
So the Campbell Lab just, by the way, you got quite a really cool bio. If you go to the Rutgers.edu website, you can see it. But recently you focused on the links between gut microbiota, brown adipose tissue. I do have a question about brown adipose. If Tony and I can still develop more brown adipose, skeletal muscle, and environmental toxicants like ozone. So, Dr. Campbell, welcome to the Sports Science Dudes.
Yes, thank you for having me, Sports Science Dudes. It's great to be here. I appreciate the invitation.
Hey, Tony and I were talking earlier. The microbiome, the microbiota, I mean, maybe, I don't know if those words are synonymous, but they're thrown around a lot.
They are.
When I was in school, and Tony and I were in school a long time ago, there was no talk about this. It didn't exist. So it's almost like all of a sudden this new field shows up. It is super complicated, but everyone's talking about good bacteria, bad bacteria. You have all this stuff in your gut, and you got to make sure you eat this, don't eat this, exercise this way. And so I guess start from the easiest thing in terms of, is there a simple way to define the microbiome?
Right.
Yeah, sure. Yeah, I mean, the reality is, microbiology is a field, the study of microbes, whether, be viruses, bacteria, so on, has actually been around for a really long time. And you can actually search back to papers way back in the day talking about the good versus the bad bugs and so on and so forth. But I think it's the extreme link lately, or intense link, I would say, to health and diet and chronic disease and exercise. I think that's really made the field explode. So I think that that's, a really great point. So the comment you made earlier about microbiome and microbiota are used a lot of times synonymously. There is like a very, minor detail. And, when you publish and put out grants, that detail does matter. The microbiota is a lot of times we say when you're talking about bugs, when you're talking about which bugs are where, it's microbiota. If you're talking about the bugs, their environment, the DNA associated with it, that's the microbiome. So just a little different. So if we talk about bugs, which we'll probably talk a lot about today, we collectively more say that's a microbiota, right? And then you want to delineate what microbiota, because a lot of environments have them, right? You have a nasal microbiota, you have an oral microbiota, you have a gut microbiota, there's a vaginal microbiota, there's a lung microbiota. People are studying the otic, the ear microbiota, and how that might be. So lots of different, so when you say bugs, you want to be specific to designate the area. And the reason I say this is because each area is rather distinct, right? The idea of your microbiota is that it works in symbiosis with the host, and that space or organ or tissue or whatnot. So, and we have tried to look at, does the lung look like the gut? And there are, of course, a few key players that are in both, but for the most part, they're very unique to that individual, tissue.
Okay, so why the focus on the gut versus, and this is something I just learned, that you're dealing with different microbiota depending on what part of the body you're talking about. So, but it seems like all people talk about is the gut. Why is
Right. So, that's a great question. I think if you're in the world of the microbiota, you hear about the various microbiomes and microbiotas and environments. But I think the reason that the gut microbiota has become such a talked about field is because of the area that all of us, the three of us, that, you know, the majority of the listeners probably that, you know, tune in here, is because it's majorly influenced by things like diet and exercise. And we know that those things are critical to healthy lifestyle and can be linked to a variety of chronic diseases. And there's no shortage of evidence linking the gut microbiota to numerous tissues outside of the gut, as well as the health of those tissues. So I think that that's why it's exploded the way it has, is because of the link. I mean, and there's a direct link between the gut and the brain via the vagus.
Mhm.
So there's a lot of talk about gut-brain axis, you know, gut mitochondria axis, gut liver axis, gut kidney axis. You'll hear probably an axis gut something or other for just about every organ, to be honest with you. And that's because the metabolites they produce, but I'm sure we'll get there at some point.
So really cool, Dr. Sara. So question quickly, it's to your point, it's very much impacted, gut particularly, by exercise, by diet. So is it the one, to the best of our knowledge, that we can most acutely or immediately impact as compared to, let's say, lung or ear, or are they all globally impacted by both the exercise and dietary practice? I would assume dietary practice, of course, would most influence the gut, but I could be incorrect because it's all new to me.
Right, right. And I think those are great questions. And this is part of where I probably would say I'm never afraid to say I don't know. I've not really looked into the literature on diet and say lung microbiome per se, right? We know that there are links between things like obesity and lung-related diseases, which, you know, could potentially have a fall back on a lifestyle, you know, sedentary behavior and or poor diet. But to say that I am completely aware of a direct link between those, I would be totally not a good Dr. Sara if I said, oh, for sure, there's got to be something, without really having thoroughly looked through the literature.
No.
If I'm being honest.
Yeah, no, great point.
Now explain, you know, when you read, when I've attempted to read papers on this, and you try to, when you look at what people write in the mainstream press in terms of the gut microbiome, or microbiota, when we're talking about bacteria, delineate or differentiate, you have good bacteria and bad bacteria. I'm not even sure how that's defined. And then also there's diversity versus, I guess, lack of gut bacteria diversity. How does that all work together?
Right. So this is always the best question. I think it generates the most exciting conversation. Because I think, you know, there is this, you know, association of, oh, certain gut bugs are good and certain bugs are bad. And in some cases, we know that there are, you know, bugs that are bad. I would say H. pylori is probably the most common gut bug that we know that are associated with a variety of gut-related chronic diseases, right?
Right.
C. difficile is another one, right? Where we're like, okay, those are predominantly going to be a pathogenic bacteria that are going to do something, you know, bad. I would say others, you know, probably get bad names, maybe not for the best reasons. You know, the best example that I can give, and I've given this example a couple of times because it really is a predominant one, is everybody talked for a while about the Akkermansia muciniphila. It's a big, you know, bug in the field that everybody's talked about, especially the diet and exercise field, because it tends to be higher in those individuals with good metabolic health, right? It's inversely associated with BMI, so on and so forth, right? Akkermansia muciniphila. Muciniphila is something that loves mucus, right? So its job is to kind of constantly refresh the mucus within the gut, which that outer loose layer we know directly interacts with the microbes because it's got the sugars and the food and all the fun stuff that keeps the microbes nice and healthy, and, you know, keeps that nice thick layer to protect the tissue. But then you go to the flip side and you look at all the literature in Crohn's disease and ulcerative colitis and colon cancer, and you find Akkermansia mucinia mucinophilia 2 to 3 times higher in
Wow.
those patients as well.
Right.
So is it a good or a bad bug? Well, if you're suffering from GI disorders, it's a bad one. It seems like if metabolically health-wise you're doing good and it's high, it seems to be beneficial, right? So I think that there is a real need in the field to go beyond labeling good bugs versus bad bugs and what's here and what's there, and really start to get at this in understanding of what those microbes do in certain environments, whether it be, why does it seem to be beneficial in metabolic health, but detrimental in, disorders where that mucus layer is really thin down, and then exposing the epithelial to this, pathogenic microbe infiltration and inflammation and immune response and so forth. So I think that activity of those microbes has become or should become important, as opposed to labeling it, and just suggesting that diversity is critical, because that's the second half of the question you asked is diversity. And more papers are coming out, and even opinion and position papers coming out suggesting that, the changes in diversity aren't actually, as prominent as we thought they were in the literature. Just as many papers are showing no changes in diversity, despite, what appears to be beneficial bugs being in that environment. So I think, again, diversity is an overused word in the field, in the absence of true understanding the activity of the microbe.
Okay. Oh,
And
Tony,
I hope that makes sense. What's
it does.
doing, right? And then you think about, how do you even measure activity, right? The best way to do it is cultivate, right? Put it in a petri dish. But then you're taking your one bug away from its community of trillions and trillions of bugs and expecting it to behave in the exact same way in your petri dish as you do in your, environment. Right. Now, don't get me wrong, cultivation studies are critical to understand how that microbe acts, what it can do, and so on and so forth. But to assume that it's going to behave similarly by itself as it does in its community with the host is a limitation. But, things we have to figure out how to get around.
Yeah. So that's like everything to your point about labeling good and bad, we do it all the time. We feel better about something because it makes life easier. This is bad, this is good. So let's just get more of this. But everything appears inevitably to turn out to be more oriented on ratio and distribution of something as opposed to good or bad, right? Like you take a look at cholesterol is bad. How can cholesterol be bad? You make it. So it's LDL, it's ratios, it's VLDL. Sodium is bad. You don't have any sodium, you live about 8 seconds without it, right? So it's the ratio of sodium to potassium that is more important than the fact that just sodium is bad. But anyway, I'm hearing this a lot, to your point, Sarah, is that people want to label one bacteria, one biota good, this is bad, you only need this one. And it's obviously more complicated and nuanced than that. And probably there's variation, I would imagine, from individual to individual, where the ratios might even be a little bit different and advantageous for each person.
Right. So, about one.
Oh, she froze.
Yeah.
Kind of complicated sometimes, right? So again, you got your 1/3 that's similar, your 2/3 that are different. And so that's, that complicates the issue. But to your point, you're talking about, individual bugs and maybe like there's groups, and, there was a term coined by a colleague at Rutgers, Li-Ping Zhao, who's one of the world leaders with regards to, gut and diet. And initially, the term was a genome interaction group, that microbes tend to work together in clusters and kind of work in concert or against other, clusters. We call these, now we kind of more associate the word guilds. So they work in guilds or groups, right? And, there is some evidence to suggest that you have, when you're good groups of microbes, and we say good again, but, in the groups of microbes who tend to promote health, are in higher abundance together. The ones that are more pathogenic tend to be in lower abundance, but there's going to be kind of this push and pull between some of those groups. And I think the critical thing is understanding, what types of microbes make up a a guild, and how that relates then to health. Because I think the idea that, one microbe is going to be the end all be all to our problems is probably
not likely.
Not likely.
Well, how many different kinds of bacteria are there in the gut?
Right. So that's a great question. So there's like 10 to the 12 or 13 like trillion bacteria down there. But ones that we can actually identify only are in the thousands. And when I mean thousands, like
only the thousands.
Yeah. The ones that we like identified, outlined, know what they do, right? So, when we run, hits, when we, extract our DNA and go through and we match those sequences with known sequences, that's what we're doing, is matching the sequences we have versus what is known to be out there, right? So the idea is you tend to see the same types and groups of microbes showing up in papers in the literature, because those are the ones that we have defined and sequenced and so on and so forth. So, the amount of microbes that aren't collectively in that group might be, a little well above what we know. And so, I think that brings you back to the diversity. So given we only know thousands and we know there's 10 to the 12 or 13 trillion, how do we not know there's functional redundancy, which is very likely, right? We know when the body fails with this isn't a backup. And, it really takes a lot for the body to really fail at something, right? You have this in place to back it up, and this in place, and this in place. And, why wouldn't that apply to the gut ecosystem, that you have several microbes that can do similar functions to ensure that that function that should be symbiotic with the host is always going on? So does that, right? So there's your diversity question. Right. So you have a lot of those things, but what if they're all doing the same thing?
Right.
That's not very healthful, right? They might be diverse, but they might all be doing the same thing. So that's where that activity gets really important to understand.
Well, let me ask you this, since we are sports science dudes, I want you to talk about, and when I read this, in my mind, I'm like, okay, that kind of makes sense, because exercise is good for everything. They say people who exercise, they got a better gut microbiome, however that's defined, than those who don't exercise. So that's the first question, why that is, what's the mechanism? Does it matter if you do aerobic exercise versus resistance exercise?
And you were going to ask me that, by the way, and I double checked the resistance training literature, and it's still, still as wah as was like a year or two ago. So major area needing to be looked at. So anyway, sorry, go ahead.
Yeah. So there's the exercise issue, and then in general, again, I'm just guessing, people who exercise also tend to eat better. So, or eat less junk, maybe. So there must be some interaction between exercise and diet versus, like me personally, and again, this is anecdote, the people who tend to eat well, all of them exercise. I have not met a person who eats well who does not exercise. So you almost can't separate the exercise and the eating well part. So could you talk a little bit about that?
Right. So, great question. So yes, the answer to your question is yes, there does appear to be a microbiota that is associated with exercise. It does appear to be a healthful microbiota. I will say what's very interesting about the microbiota, and I put this out there a couple of years ago, that it is like what you would consider a training effect, because there is evidence to suggest that you exercise and it alters. There are even studies out there to suggest that microbiota will change depending on the part of season you're in, whether it be a preseason versus the postseason versus intense training versus not intense training. That microbiota will adjust every single time. But there is also evidence to show that when you stop exercising, that benefit goes away. similar to what we think
Energy.
about with training
Yep.
adaptations, right?
Yep.
If you stop running, your VO2 max will decline.
Okay.
It needs that constant stimulus.
So let's go back to you mentioned there's a benefit that may disappear when you stop training.
Right.
What is the clinical manifestation of that benefit?
So I think that's the complicated question, and what the 3 grants I have in NIH right now that hopefully they'll fund me on are meant to look at, right?
Okay.
So the number of studies out there that initially came out, including ours in 2016, was like, there seems to be this overt benefit with butyrate producing microbes, increasing butyrate as a result of exercise. Butyrate is a short chain fatty acid along with acetate and propionate. And butyrate is definitely known to enhance colonic health, right? Increase cell proliferation, differentiation. And given this, turnover in this healthy interaction, we know that there's a lot of immune, right, system cells within the gut. We know we have both mucus and gut-associated lymphoid tissues, the malts and the galts that are in the gut. So, keeping that intestinal lining healthy, that turnover regular, kind of keeps the balance too of the inflammation responses and the immune responses, right? So, if that's the case, you have this butyrate and it's making these, healthy colon, chances are you're not overtly enacting an immune or an inflammatory response, which we know can manifest itself into a cold or a disease or, something else. So as the literature has evolved, there have been a couple of studies, both animal and human. I think one of the big ones that made it out was a couple of years ago when Scheinman and colleagues looked at the Boston Marathoners. Oh. And had this study of 10 individuals roughly who completed the Boston Marathon and found a microbe that was very unique to those runners, Villanella atypica, which, now has become pretty popular. And so they did a series of very elegant studies to really look at that mechanism and found that it was, through a propionate pathway, right? And how it managed lactate, which in some ways, for makes marathoners and training at that lactate threshold can be really important. Yep. So, okay, so now you have the early studies talking about butyrate. Now you have, some distance runners talking about propionate and a lactate connection. And they took this Villanella and put it in a germ-free mouse, meaning a mouse with no microbes, just to see what that microbe in and of itself did to the mouse. And it did run, and it did, demonstrate and display some of those properties of that, lactate and that, and that mechanism. So that's one. More recently in an animal study, we see acetate playing a bigger role than butyrate and or propionate. So I think, some of the working, hypotheses and things that we're thinking about are, is it possible that exercise has several of these guilds that are responsible for increasing exercise performance, right? Mm-hmm. Lactate might have its own set of microbes that do something to help it, right? There was some studies where we used what they call notobiotic mice, which is a mouse with a known set of bacteria in there. Some were good at altering, lactate dehydrogenase, some changed creatine kinase, but not all of them had an effect on every outcome they measured, suggesting some are going to be good for some things and some are going to be good for other things. So the reality is, right, there's probably several different groups of microbes that regulate a variety of, of interactions. And we know the microbiota is important because, there's, more papers coming out, including ours, on this bidirectional link. So it's not just what does exercise do to a microbiota, but what does the microbiota do for exercise? So when you knock out the gut microbiota, there are these impacts on exercise, right? And so I think that's one of the ways to look at mechanisms. When you knock out the gut microbiota, what about exercise changes? Mm-hmm. And does that tell us the microbiota might have a link to that aspect of physiology then potentially?
And performance. That's amazing. I mean, you talk about the
That help,
global
does
adaptive.
that answer your question though? I mean, I know,
Yeah,
I think it's a loaded question. I think that's the big thing. I think the bottom line is the mechanisms are not entirely clear. I think people are devising ways to figure that out. You know, there's a couple on those, like I said, microbiota knockout, I should say, or, you know, knockdown using antibiotics that have uncovered some really cool things recently. So we can talk about that if you're interested as well.
No, this is, I mean, this is definitely a field where it seems like you're peeling, you know,
Oh yeah.
umpteen layers of an onion. And the more you learn, the more you realize, wow, it's just not very simple. I mean, it's almost like studying the brain in a sense, because you can't biopsy the brain, right? You
Right.
can't really, I
Right.
guess, could you get gut bacteria out of someone without being I mean, you'd have to be invasive, right? I mean, I
No,
don't know.
honestly, they just kind of poop in a something for you.
Okay.
And you just, you know, put it and you do the extractions and so on and so forth. You know, the flip side is a little bit more interesting just because, you know, we're actually in the middle of this right now. So kind of cool. So I don't feel bad about, you know, saying things, but you know, animals, we can give a cocktail of antibiotics to really eliminate the microbes.
Interesting. Yeah.
Right? Get at, you know, it's a cocktail of a whole bunch of broad spectrum, gram negative, gram positive, anaerobe, aerobe, try and get rid of everything you can. I can't really do that ethically in a human. Hey, take this ginormous cocktail and like, you're going to have no microbiota. So let's see how it goes. Right. So we can't do that. But what we can do, and what we are doing right now at Rutgers, and, you know, one of my PhD students is doing this for his dissertation, and we're doing it with this, you know, a collaborator in Colorado. So it's a multi-site, but we are taking basically a Z-Pak, probably the most commonly prescribed med, probably one of the most commonly prescribed meds, you know, among athletes.
Yep.
And giving, you know, athletes in the absence of a cold, right, because that's going to confound your data, a Z-Pak for a week, and testing them before and after, and get fecal samples and blood samples before and after to look at serum metabolomes and microbiota changes, and is there change in performance. And, you know, we figure, you know, I'm not 100% sure a Z-Pak is enough truly maybe to mess with performance.
Right.
Will we probably see changes to bugs? Probably. But, you know, we're not sure it's strong enough. Either way, we feel like it's okay, because, okay, then we say, well, the Z-Pak maybe changed microbiota, but not enough to impact performance. So it's still a safe antibiotic to give your athletes, right, when they're ill. You know, if it does change both microbiota, metabolome, performance, then we go back to the drawing board and say, all right, we may need to exercise some caution when giving athletes a Z-Pak, especially in the middle of the season. So, you know, we really want to make sure you get a full translation of the things that we're seeing with, you know, animal models in the human model, and to what extent you can replicate some of that.
So that's phenomenal. Quick question on that. Independent of putting performance aside, can something like how long is a typical Z-Pak there, you know, last? How long are we taking that if we did have a, you know, a reason for that? About a week, 2 weeks, or?
Right. So it lasts about that long. But a Z-Pak is really a 5-day course of antibiotics.
Yeah.
It's usually the 2 pills on the first day, 1 pill on each additional day for 5 days. But it's a longer acting antibiotic, so it's supposed to stay in your system, you know, for the 1 to 2 weeks, just to make sure it kind of kicks everything out of the way that it needs to. So we're trying to get the participants within 24 hours of taking that last dose to get them back in the lab, just to, you know, check. So it's more at the height of when that medication might be, you know, rather than the 2 week, you know, later, just for consistency reasons, you know, controlling for the study and all that good stuff.
But something such as the Z-Pak could make again, regardless, or put performance aside, a very substantive change in a very measurable change in microbiota. Then you're saying, right? It
could, yeah. And you know, and which microbes, you know, it's meant to be a broad spectrum. Will it knock out, you know? The question is, in the absence of truly knowing which microbes overtly benefit exercise performance, do we know that it's knocking it out? I think still in the absence of knowing what microbes overtly affect exercise performance, taking that antibiotic does, mess with performance, that just strengthens the argument that there's a microbiota link to exercise.
Right.
Could you address the both animal and I'm not familiar if there's much human data on fecal transplants and how that affects health?
Yeah. So that's an outstanding question. So recently, we know the FDA kind of give its little approval for fecal transplants for things like C. diff. And so, because that's very common and they appear to be, effective for C. diff. So fecal transplants in animals, let's start there and we can go to humans, can be really interesting. You can actually do the fecal transplant where you take the feces from, say, your exercised animal, which, Woods's group did, kind of put it in its little solution and kind of, provide that transplant to another set of animals. And, his data very nicely showed that, you can take exercised microbiota or fecal samples, give it to non-exercised animals and see if it protects against DSS-induced colitis. And it appears to have worked in that way, right? There are other studies, because we always get the question, well, if you take the microbiota and you give it back, do the animals resume their exercise performance, right? To kind of complete that circle. Right. And, rather than giving a transplant, what they did is they did what's called natural reseeding. So mice are coprophagic, so they'll eat poop. So you basically take the exercise poop and just kind of put it back into the cage. And they did that for about 10 or 12 days. I think 10 days after, and even after, so they gave them about 21 days of antibiotics. Yes, found the reduction in exercise that, many of us did, refed them, reseeded them, I should say, for 10 days. Exercise seemed to have come back. And so, it just kind of completes the loop, makes that evidence a little bit stronger that it does that. So there is that aspect of, a fecal transplant in that, humans, we don't do that. So it is really, a more of a cocktail of things. And like I said, for C. diff, it seems to work. The one important thing to note is the microbiota, as I mentioned, as an exercise adaptation. If you stop doing it, sometimes those microbes will revert. So that fecal transplant, you have to be sure that it takes, right? And so there are some studies to suggest that in some individuals it takes and it stays much longer than other individuals, which makes sense, again, given that inter-individual variability. So, it does appear to be helpful with disease. I don't think we've quite gotten there to, say, give a marathon, a non-exerciser, a marathoner fecal transplant, and just be like, hey, go run a marathon. We haven't quite got there yet. So
what about the data on body fat or obesity if you do the fecal transplant?
So yeah, some of that is really interesting. I think the best study came out of Jeff Gordon's lab, the discordant twin study. That's one of the big ones that you're talking about, where they kind of took the obese twin and the healthy twin and they switched their microbiotas, and yes, phenotypes started to match of the donor microbiota. So
it's insane.
It really is.
But, and I think that studies like that are why, and you loop back to your very first question, why has it exploded so much? And it's just, some of these, studies have really shown that it makes a big difference what that microbiota is and how it can help foster that healthy or unhealthy phenotype.
Right. So
go ahead, Tony.
No, it's just phenomenal because, the interrelationships with every system are probably far more than we ever thought, right? And now we're finding this out about the gut. And I don't want to move off to a completely separate topic, Joe, if you have more questions related on the exercise side. But like everything else,
I have more answers for that too, so we can't get off it too much yet. I'm just kidding.
Like everything else, Sarah, I think what we're seeing now is the gut is being associated globally with every either illness or everything that's good for you, right? And that's
Right.
what we tend to do also. We don't just say this is good or bad. Now we found the holy grail to every other health ailment. But the link, what do we think we know about mood and gut? So I was even looking at some reading this morning, and many people now are attributing, even if it's a clinical depression, I'm not talking about dysthymia or a low-grade chronic depression, but something that on a Hamilton or PALMS would be related as clinical, and saying, well, that is directly impacted by the microbiota too now, and the gut,
Right.
and stuff like that. Do we know if there is a relationship, and if so, what mechanisms are going on there? I mean, how is mood being impacted? Is it through neurotransmitters? Do we know anything about this excessive serotonin production or anything like that? Or
Right. So those are all great questions. And so just remind me to get back to the hypertrophy and gut microbiome elimination.
we could go to that first. Go to
Yeah,
that.
no, studies and like, we just did the mitochondria study. So
Do
some
that
really
first.
interesting things going. No, well, we can, it's okay. Your point though is, I think, people tend to like say now the microbiota is everything.
Yes.
Change the microbiota and you'll be automatically healthy. And I
Right,
think for those of us physiologists who know how, both interactive but independent systems can be sometimes, like it's,
right.
It's always that if it's too good to be true or it sounds too simple to be, it probably is, right? And the microbiota, isn't going to fix everything. Are there links between a lot of them? Of course. Is there a link between the gut and the brain, as I suggested? Yes. It's probably one of the most well studied, to be honest with you, because of that connection via the vagus nerve, because of the, known microbial derived metabolites that are found in systemic circulation that have the potential to cross the blood-brain barrier, right? And exert some influence there.
Okay.
And so I think that, those are really interesting. I was just happening to be, PubMeding, right? Because our world, we don't Google, we're good, we PubMed. Like I was PubMeding the other day, and there was some really interesting, article that I didn't get to read yet, but talking about microbes being linked to exercise motivation in the brain, right? So microbes and potential metabolites that can be linked to areas in the brain responsible for motivation to exercise, right? There was a study, because I pay close attention to the estrogen and stuff going on, since mine is like waning as the, whatever these days. It's so fun. But, there's been links between like, estrogen and lack of those receptors in the brain and motivation to exercise. And can you find a way to enhance some of that with, gut and things? So there's no shortage of those, studies that are out there. Not my primary area in terms of gut brain, but these links are certainly there. There are PubMed and published articles in this area suggesting that there's this very real link, especially due to the microbial derived metabolites, which is a lot of what, secondary bile acids. And so, a lot of that going on in terms of the main way that we think the microbiota can act outside of itself.
Yeah, okay, phenomenal. So it is pouring enough into systemic circulation to potentially impact that, as you noted. Yep.
Yeah, so that's
really
that's one of the important things about measuring like serum metabolomes is, what microbes, or what metabolites are now in system to then be perpetuating these effects. So yeah.
Incredible.
Yeah.
I have a question about the when you look at the way people train, because I want to get back to the resistance training part.
Yeah.
But there are a lot of people who sort of do a combination of everything. They're not just pure aerobic creatures or pure resistance training. I mean, a lot of us kind of do a little bit of everything. So does that mean the adaptive response of the microbiota is sort of a little bit of everything? Or is there a unique response to pure aerobic training versus pure, let's call it hypertrophic or bodybuilding type training?
Right. And so that's a great question. So there does seem to be, because most of the studies, as I mentioned, in PubMed, I was like weightlifting, gut microbiota, resistant training, gut microbiota.
3?
Wow.
Wow. Okay.
Weight training and gut microbiota, there was 2. For whatever reason, resistance picked up another one, but it's like 2-3 articles, legitimately. And one was really interesting. It was like some, I might actually, oh, I won't go away, right? So the one I did find, confounded, I say confounded, but some of what they were looking at, because it was like eating a high protein diet, right? So,
That's a confounder. I mean,
right. So is it right? And then the other one was the ergogenic properties of a bifidobacterium longum isolated from a weightlifting gold medalist. Both studies were in nutrients, and both 2019 and 2021. So just very recently. And those are the only two for me that pop up when I do weightlifting and gut microbiota.
Wow.
So to suggest that there's a unique weightlifting or resistance training microbiota is probably far from being elucidated. There's, like I said, only those handful. I mean, I'm sure, PubMed gets everything, but there are sometimes things, I mean, if I were to go to Google, and probably something else might pop up, but the reality is it's not as well established as the aerobic side of things, right? Most of it's been in aerobic exercise. Most of it does suggest that, yes, there's a microbiota that appears to be beneficial and healthful as a result of aerobic training. And this is consistent in males, females, animal, and human studies.
So there's no sex difference. There's at all.
No, I didn't say that. I said it does appear that there are beneficial changes in both sexes, but those changes are definitely a bit different. Oh. So the microbiota within the males and females are different to begin with, right? And short chain fatty acid profiles. Okay. So we are currently in revision on a sex difference and time course study on sex, diet, and exercise on gut microbiota changes and the impacts. And there are definitely pretty prevalent sex differences that are abundant. Yeah.
What about old versus young guy differences, since Tony and I are on the older side?
Right. So good question. This was a study, and I believe it was rats. Mika published it. It was at Moni Fleischner's lab out at Colorado. And they looked at exercise initiation and alterations in gut microbiota in young versus old, right? Appeared to be changes, definitely more robust in a young animal compared to the old animal. But that doesn't necessarily account for, which I assume all three of us are lifelongers, right? Mm-hmm. I mean, we've started exercising when we were younger and short of periods of time where, where some of us maybe have been very sick or so, kind of kept up a pretty regular exercise routine. So does that mean that exercise manifested changes that have been fairly salient through our aging? I think that that's a great question, and probably one that hasn't been answered yet.
It has in many other systems. So, I mean, we don't know, but it might be rational to assert that it has, it could preserve
right.
it well.
Right. Yeah. If you start exercising earlier, you benefit from those adaptations that maybe makes a more resilient gut microbiota even as you age. Yeah. I mean, that is a reasonable hypothesis for sure.
Okay. Delving into the supplement side, tell us a little bit about probiotics. Are they worthwhile, worthless, and also the prebiotic category? So talk a little bit about that.
Yeah. So, the pre and the probiotics. So the probiotics are the bugs themselves, right? You tend to take them. The prebiotics are the foods that, fermented kind of things that go in there and get made into those short chain fatty acids. Probiotics, I am when it comes to supplements, I am a do no harm, right? So, and probiotics definitely do no harm.
Okay.
So I think every person you talk to in a probiotic will tell you their story, or an ex-user of a probiotic. They either totally work for you or they don't at all work for you, right? I think, probiotics are, again, once you start taking something to change the gut microbiota, you have to take it. And when you remove that stimulus, the gut microbiota will revert to what it's comfortable at which is your microbiota that gets kind of settled in at 2 or 3 and then has changes throughout the life. Puberty will induce something and this will induce something else and so on and so forth. But it tends to kind of stay fairly right? So if you start the probiotics, keep taking them, especially if they work for you. If you take them and you stop and you don't notice anything, then perhaps it hasn't done much, right? I mean, most of the probiotics I still think contain very similar still versions of lactobacillus and bifidobacterium. I don't think that they have kind of moved too far away from a lot of those, strains, because they tend to be the ones that we know the most about. But given the earlier parts of our conversation where we said, well, we only really know how a few thousand work, right? Are there definitely untapped ones that we don't know about that could be more important, and so probably, maybe, right? And they just, haven't been looked at yet. I think prebiotics are, again, that's again linked to your diet. Are you incorporating those, high fiber foods or fibrous foods as part of your diet that will then manifest the production of those short chain fatty acids? And again, if you're eating them and you notice good changes to your gut microbiota, don't stop, because once you stop, it'll go back. And, you unfortunately see this kind of like reverse, or reversion of the gut microbiota to whatever it was after you stop or give up whatever that intervening aspect is. So, most people will say feed the bugs, the prebiotics, use the probiotics if, you are deficient, maybe have some sort of gut related issue that, you feel benefits them. I think most people would say kind of like feed the bugs. And we know for performance, you guys, I believe you guys have you have a position stand on probiotics where it's not really, it's kind of not direct, it's an indirect ergogenic on maybe, potentially kind of lessening sick time and things like that, just because I think, the mechanism of how probiotics work is still totally unclear. I mean, for a while there, the NIH was putting a lot of funding into how do probiotics work because everybody seems to be on them, and gut microbiota is big, and so let's figure out how they work. And so, it says something that within the last 10 years there were major calls for proposals of the NIH to understand how they work.
Right. So one quick question, and it's just hard to answer, I know, so without taking direct measurement. You said some people take them and think it works, right? What are we looking for? What would you, if outside of direct measurement of what's available, what do we mean by work? And I'm not being a wise guy. Like, what would we
totally
look for, right?
Yeah.
Like,
yeah, no,
what
absolutely.
would that mean?
Right. So short of, like you said, checking to see what that gut microbiota looks like before and say after your probiotics, and were you deficient prior to in some of those probiotic bugs, which we do know are beneficial, which is why they're put into the probiotics. And now they're there acting as a guild, suppressing pathogenic guilds, right? In that whole interaction that we were talking about. So you don't know that, and like you said, in direct measurement. I think the biggest way to see if they work for you is, alleviation of symptoms. For someone, say, suffering, someone feels bloated or crampy or whatever all the time. I think the biggest thing is, do your symptoms feel alleviated to where the comfort of your day is a lot better. And you're like, okay, well, if you're noticing that, then you probably are like, okay, these are probably working for me.
Got it.
Does that make sense?
Yeah, totally.
Yeah.
We're almost out of time. I want to be respectful of your time. So really one last question, and it's a pragmatic one. So if someone were to ask you, what are the things I can do in terms of diet, supplementation, and exercise to have a healthy gut microbiome, you would, and assume they know nothing about science.
Yeah.
What would you tell them to do?
Right. So great question. So based on the literature available at the moment, right, engaging in at least some aspects of aerobic exercise, so at least starting with this something like, the 30 minutes, 5 days a week, and, working your way up, but engaging in aerobic exercise, because we do know that's beneficial in both men and women who exercise, right? Diet wise, I think there are probably lots of diets that haven't been looked at with regards to the gut microbiota. So, saying like, oh, high carb, low fat, high, but we do know, those prebiotic type foods that facilitate the activity of those microbes that are going to be responsible for producing short chain fatty acids and other metabolites are typically assumed to be more healthy. That doesn't mean you don't eat probiotic type foods like, the yogurts and the, all of that good stuff as well. In terms of, supplements, I think that's probably also really understudied area in terms of what does, and I probably could PubMed it, or one of you, like creatine and gut microbiota. What does creatine? And we know that that's extremely beneficial for performance and a variety of other things. But what does that do to your gut microbiota? I don't know that we know that yet.
That's a
right?
Great question.
Right? So maybe I smell a study brewing, guys.
Yeah, there we go.
Or dudes, however we want to say it. But yeah. But I think probiotics is probably the biggest, supplement that's out there. And again, that's really a very, personal thing. If it benefits you, great. If it doesn't. But I think the biggest thing is, for sure, we know aerobic exercise helps in both men and women. For sure, we know the soluble, insoluble fiber type diets are beneficial for those microbes as well. I think a lot of stuff outside, and I should say, and we know high fat diets are typically bad.
Okay.
Okay.
Right. So,
I should keep eating my kimchi and occasional sauerkraut.
Yes, absolutely. Why not? It's delicious as well. So might as well enjoy a little bit of life, right?
Right. Any final thoughts, Tony, for Dr. Sara?
Fascinating. I mean, it's just an incredible, it's a universe, right? You're talking trillions. I mean, I don't know, what are there, trillions of galaxies out there. You're talking numbers that you can't quite fathom. And it's really exciting. You gave us a great overview, Sara. I thank you. And I'm really looking forward to what the field will tell us in this discipline in the future and your work, because it's in infancy. And when you're talking about trillions of cells, or I should say trillions of, those organisms, boy, there's a lot to learn and a lot going on here. So we got a fun future ahead,
Yes, definitely.
Sure. Well, thank you, Dr. Campbell. I mean, this was quite the edification. I learned a lot more about microbiota and the microbiome than I have in the previous 5 or 10 years. So appreciate your time, and thanks for being on the Sport Science Dudes.
Yes, well, thank you so much for having me. It was a blast. I appreciate it.
This was great. Thank you again.
All right, thanks.
Anytime.